Development usually occurs at the 4th week and it appears in the form of limb buds. The limb buds consist of a core of mesenchyme derived from the lateral plate of the somatic mesoderm and an outer covering of the ectodermal cells. We already know that the mesoderm consists of the paraxial, the intermediate, and the lateral plate of mesoderm. The lateral plate of mesoderm actually gives rise to the core of mesenchyme forming the limb buds. Now this core of mesenchyme is covered by the apical ectodermal ridge, which is induced by the fibroblast growth factor. The figure shows the embryo with the limb buds, a forelimb bud and the hind limb bud. The forelimb bud appears a bit earlier, about 2 to 3 days earlier than the hind limb bud. This is a mouse model showing you the limb development of the forelimb development and the hind limb development. The forelimb, slowly and gradually, the mesenchyme has changed, is taken over by the formation of the cartilage, and then slowly and gradually the cartilaginous model is replaced by the bone. The upper limb buds arise a few days before the lower limb buds. The upper limb buds develop at the lower cervical and the upper thoracic levels. The lower limb buds develop at the lumbar and the upper sacral regions. This is a figure showing you the limb buds, the appearance of the limb buds and the presence of the mesenchyme, and slowly and gradually the mesenchymal proliferation takes place throughout the limb, and this mesenchymal proliferation distally the cartilage takes over it, and slowly proximally the cartilaginous formation occurs and it is taken over distally later. This shows the cartilaginous formation, and slowly and gradually the cartilages appear in the digits as well. The mesodermal cells closest to the apical ectodermal ridge proliferate, and as this proliferation continues, the cells further from the mesoderm begin to differentiate. As I told you earlier, there's a formation of the mesenchyme. The mesenchymal cells are proliferating towards the apical ectodermal ridge, and this proliferation continues, but further away from the apical ectodermal ridge, this mesenchyme differentiates and it forms the cartilage. Mesenchymal cells aggregate throughout the cores of the elongating limb buds to form the limb skeleton. Then the chondrification centers appear and the entire limb skeleton gets cartilaginous. This figure shows the mesenchymal appearance, the mesenchymal formation from the lateral plate of mesoderm, and then this is taken over by the cartilage, which appears distally first and then proximally later. Slowly and gradually the entire limb gets cartilaginous. The osteogenesis, mostly by the endochondral process, begins within the clavicle and continues throughout the fetal life and the childhood. The primary ossification centers start in the shaft of the long bones. An epiphyseal plate of cartilage occurs between the primary and the secondary ossification centers. First, there's the presence of the mesenchyme. Mesenchyme is then replaced by the cartilaginous appearance. Cartilaginous model is formed, and then later on the bone formation occurs, and it begins with the clavicle. Now, when the bone formation occurs, there is a primary ossification center that is formed in the shaft of the long bones, and there is an epiphyseal plate that occurs between the primary and the secondary ossification centers. Here is a figure showing you the mesenchyme. The mesenchyme has been replaced by the cartilaginous model, and later on the cartilaginous model slowly gradually replaced by the bone formation. You can see over here the bone formation, the primary ossification center has started, and it has this appearance of bone formation, and slowly and gradually a secondary ossification center appears at the epiphyses, and they also start to form the bone, and what remains in the center is the epiphyseal growth plate between the primary and the secondary ossification centers. Now, as the limbs develop, there's a development of the appendages as well, that is the hand and the foot. The distal end of each limb bud becomes flattened as the pedal-shaped limb plates. These are the primordia of the hands and the feet, and the development is also induced by the apical ectodermal ridge. So each limb bed becomes flattened as a pedal-shaped limb plates. Here we can you can see the figure how the embryo has the limb buds, the upper limb bud and the lower limb buds, and initially it is a plate-like appearance, and slowly and gradually this bud develops certain ridges, mesenchymal ridges, and these mesenchymal ridges are covered by apical ectodermal ridge. There is an ectoderm there that induces the formation of these digital rays, which are the future fingers and the toes in the lower limb. The excess tissue, the mesenchymal tissue between the digital rays, is removed by apoptosis. Apoptosis is programmed cell death. That is, the excess tissue between the digits is degraded or degenerated by the formation of the programmed cell death. And so we have individual fingers and defined fingers and toes. If this programmed cell death doesn't place, then there is a fusion of the fingers which gives rise to anomalies such as the syndactyly, in which the fingers have not been separated and there is a tissue present between the fingers. At times there are excess, usually we have 5 fingers and 5 toes. At times there is either there's an absence of a digit or there may be an extra digit presence. In case of an extra digit, we call this anomaly as polydactyly. That is, an extra digit is present in it. 5 mesenchymal condensations, the digital rays appear in each plate. They eventually quadrify and ossify to form the long bones of the hand and the feet. That is, the metacarpals, the metatarsals, and the phalanges. We initially said we had the buds, we have the plates, mesenchymal plates, and slowly and gradually there are 5 digital rays that appear within these buds, limb buds, and then these limb buds, the mesenchymal structure changes into a cartilaginous form, and then slowly and gradually they ossify, and eventually we have the long bones of the hand and the feet. The loose mesenchyme lying between the digital rays eventually degenerates with the overlying ectoderm, leaving interdigital clefts and thus delimiting and defining the fingers and the toes. So initially those digital rays that had appeared, they had the mesenchyme in between that gets degenerated by the programmed cell death or the apoptosis. Now coming to, we have seen how the limbs have been developing. There was development of the mesenchyme by the apical, under the induction of the apical ectodermal ridge, and then slowly and gradually the mesenchyme has changed into cartilage, and the cartilage gets ossified, and then we have the rotation of these limbs. We have the rotation of these limbs, and during this rotation takes place at about the 7th week. The upper limb rotates about 90 degrees laterally, and the lower limb rotates 90 degrees medially. Initially, these the two limbs rotate in an opposite direction. That is, the upper limb rotates laterally and the lower limb rotates medially, so that the extensor muscles lying on the posterior surface of the upper limb and the thumbs also come to lie laterally. And the lower limbs rotate 90-degree medially, so that the extensor muscles that come to lie on the anterior surface of the lower limb, and the big toe lies on the medial side, and the thumb lies on the lateral side. This is a figure showing you the sequential rotation of the limbs. Initially, the limbs are extending ventrally earlier in the 7th week. The developing upper and lower limbs rotate in opposite directions and to different degrees. The upper limb rotates laterally 90 degrees on the longitudinal axis, so the future elbows point dorsally, and the extensor muscles lie on the lateral and the posterior aspects of the limb. The lower limbs rotate medially through 90 degrees, so the future knees face ventrally and the extensor muscles lie on the anterior aspect of the lower limb. You can see the limbs over here in the embryo. Initially, they are lying ventrally. Slowly and gradually, the upper limb buds rotates laterally at about 90 degrees on its longitudinal axis, and it rotates in such a way that the elbows will be facing posteriorly. And the extensor muscles lie on the lateral and the posterior aspects of the limb. Whereas in the lower limb, though it was initially facing ventrally, but it has moved medially about 90 degrees, so the future knees face ventrally and extensor muscles lie on the anterior aspect of the lower limb. Now coming to its various deformities. As far as the rotation is concerned, if proper rotation doesn't take place, then the limbs stay in the same state as they were in the embryological stage, and that's why it will give rise to difficulty in the movements of the limb. Now coming to the deformities of the limbs, we already talked about polydactyly, which is more than 5 digits. Normally we have 5 digits. In this figure, we can see about more than 5 digits, about 6 digits present, and these occurred because of the extra digit being formed, and that extra digit formed has led to polydactyly. Syndactyly is a condition in which the extra tissue that was present between the digits was not degraded, apoptosis didn't take place, and the digits are fused together, and a surgical maneuver has to be done in order to separate these digits. Then there are other anomalies such as Talipes equinovarus, and in which the sole of the foot is inward with the foot adducted and the plantar flexed. That is, the foot has been medially moved inside, and that is called as the club foot or the Talipes equinovarus. Syndactyly, again the same thing, where there is no separation of the digits, and then there is a lobster claw formation in which there is a failure of the middle digital ray to form with absence of middle metacarpal or metatarsal and middle digit, and there is a deep central cleft. Here we can see the syndactyly, where the digits have been fused. The lobster claw hand, see, we said there is an absence of the middle digits, and a large cleft has been formed. It resembles that of the lobster, and only 2 digits are present. Then there is another abnormality which we call as the amelia and the meromelia. Amelia is when there is complete absence of the limb that is, the limb bud did not form, and from the lateral plate mesoderm, and there's a complete absence of the limb. Meromelia is a condition where the digits are where the limb is present, but only a little part of the limb is present. Complete limb is not present. There are several factors contributing to these defects. There may be environmental agents, they may be genetic, or they may be both. Here we have shown you the Talipes equinovarus. There's another condition where in this the limb has been turned inward and it has been moved inward, and if it is noticed initially, then it could be through certain measures. It could be using certain shoes and certain measures, certain plaster surgeries, it could be corrected. But if it is not observed initially in the initial stages, then there would be later on surgeries would have to be done. And then another condition, there's Talipes equinovalgus, where the limb is here, it is moved medially, but in that the limb is turned laterally. The foot is turned laterally. Thank you very much.